Dear students, today we are going to study the structural and functional organization of bacteria. Bacteria, as you all know, they belong to prokaryote group. That means they do not have a perfect kind of nucleus and their nuclear material and their genome remains unsurrounded by any kind of membrane. Let us come to first of all, let us study the size. The smallest bacteria which we come across, they are of 0.1 micron diameter, and the largest they are about 60 into 6 microns in dimension. The smallest bacteria are Rickettsia and Chlamydia, whereas the largest is Spirillum volutans. The shapes of bacteria depends on the plane of their division, and we have four kinds of shapes in the kingdom of bacteria. First one is spherical. They are called as cocci. Among the cocci we have Diplococcus, Tetracoccus, Staphylococcus. The Staphylococcus are plate-like. The Diplococcus, they have two cocci, and Tetracoccus having four spherical cells. Streptococcus, they are chain-like, whereas Sarsinus, they give impression of ball, solid ball. Second shape is rod, and in this category we have Bacillus, and it gives an appearance of a small rod. Third shape is spiral, and the example is spirilla, and as depicted in the picture, it looks like spherical. It looks like a spiral rod. Mycelial forms mycelia like structure, and the example is actinomycetes. Now come to this diagram which is pictured in the plate. Afterwards we'll take up the theory part. Let us see this diagram, and let us move from outside in. At the outside you can sight this flagellum, which is present at the topmost position and arising out of blepharoplast. Then we have capsule. The outer is microcapsule and inner one is outer is macro, inner is microcapsule, and after the cell wall we have this plasma membrane, and this cell wall. After the capsule we have cell wall. You can see this cell wall of green color in the diagram. The cell wall is made up of proteins, lipids, polysaccharides, lipopolysaccharides, and peptidoglycan. Depending upon the composition of the cell wall, the bacteria have been divided into two classes, gram positive and gram negative. We'll study separately the differences between gram negative and gram positive bacteria, and the difference is created by wall itself. Then if you move inside the cytoplasm of bacteria, you can see some inclusion bodies which contains lipid, starch, metapoliphosphates, metapolyphosphate sulfur. Then one thing I would like to remind you is that they don't have any membrane bound organelles. The membrane inclusion or inclusion bodies, they have a separate kind of membrane which is somewhat different from the unit membrane concept. At the topmost position in the blue colored plasma membrane, you can see mesosome, which is the in pushing of plasma membrane inside the cytosol of the bacteria. Then we have this nucleoid which is attached to the plasma membrane. This nucleoid is not surrounded by any nuclear membrane, which is the characteristic feature of the prokaryotes. It is laid naked in the cytoplasm but remain attached to the plasma membrane. The mesosome is also a very important structure in the structure of bacteria, in the sense that it serves the purpose of mitochondria. All the electron transport activities they are performed by the mesosomes. Only organelle which is present in the bacterial cytoplasm is ribosome, and that ribosome has 70 S Swedberg units. Otherwise, you won't come across any membrane-bound organelles starting from the nuclear membrane, endoplasmic reticulum, Golgi body, lysosome. Nothing will be seen in the bacterial cytoplasm. So, you can see in the picture that this is the cytoplasm with ribosomes, and the loose slime is part of this capsule. Then we have this granules of poly beta hydroxybutyric acid. They are also membrane bound, but the membrane has a different composition. So this is about the structure of the bacteria. Now let us come to some theory part. In the structure, let us see what is there in the cytoplasm. So the cytoplasm of bacteria contains enzymes, coenzymes, then vitamins, metabolites, inorganic ions, cell inclusions like lipids, starch, metapolyphosphate, sulfur, and carbohydrates, proteins, and fats are the usual components. Then comes ribosomes. As I have told you, you come across 70 Swedberg type of ribosomes in the bacteria which belongs to prokaryota, whereas the eukaryotes, as you know, they have 80S type of ribosomes. They occur in form of polyribosomes, means forming bead-like structure with the RNA. The internal membrane system, as I have told you, is absent. The protein synthetic mechanism of bacteria is hampered by two drugs, namely streptomycin and chloramphenicol. These two antibiotics, they target the protein synthetic mechanism of bacteria, and therefore the bacteria, they cannot synthesize any material like cell wall and all that, so they get died. Now the cytoplasm has some food storage granules which are membrane bound and also called as inclusion bodies. In the beginning I told you that the bacteria, they don't have any membrane bound structure. So this membrane bound food storage granules, they have different kind of membrane which is made up of lipid bilayer, and the composition is different from that of plasma membrane, or they may be part of in-pushing of the plasma membrane. The food storage granules are glycogen granules, the carboxysomes, the chlorosomes, and magnetosomes. The membraneless granules are also cited in the cytoplasm of this bacteria, and the membraneless granules are volutin granules, polyglucan granules, poly-beta hydroxybutyrate granules, which is the food reserve in absence of nitrogen. Some bacteria, they photosynthesize, and the unit of their photosynthetic apparatus is thylakoids. Now, their pigments is contained in chromatophores, and these are also membrane-bound organelles. Excuse me, vesicles, which are about 300 angstrom in diameter and contains pigments and enzymes necessary for photosynthesis. For instance, cytochromes, carotenoids, and in the carotenoids the bacteria have bacteriochlorophyll, which is a deviation from the normal chlorophyll which we have in the higher plants, and it also perform the it also contains some electron transport enzymes and ferredoxins. The ultimate electron acceptor in the photosynthesis of done by bacteria is other than H2O. So whenever bacteria they photosynthesize, they never produce oxygen. Now nucleoid. What is this nucleoid? Nucleoid is their genomic material which is present and not present in the nuclear membrane, or not surrounded by the nuclear membrane. It has about 100 micrometer diameter, and when it is cut open, it extends from 1 mm length to 13 mm length. It is highly folded and is remain attached to the plasma membrane. But one thing has to be noticed here, that his nucleoid doesn't contain any histone, unlike eukaryotes which have histone protein surrounding their DNA or genome. But instead of histone proteins, the bacteria they have polyamines in place, and they stabilize, they form a stable structure with the interaction while interacting with the nucleic acid. The DNA of nucleoid also contains some methylated bases like 5-methyl cytosine and 6-methyl aminopurine, etc. Another very unique kind of structure which is present in the cytosol of bacteria, they are the plasmids. Plasmids are actually the circular DNA molecules which self-replicates by themselves, and they are regarded as the extra genetic determinants, and they are also made up of DNA. They are discovered by Jacob and Holman, and in fact they contain some antibiotic resistant genes, and the plasmids they are not necessary for the survival of the bacteria. But one important thing they do to the bacteria is they confer the sexual dimorphism or maleness to the bacteria in which they are present. The bacteria they are also called as F episomes and are responsible for maleness or sexual dimorphism. At times when the plasmids they just bind to the nucleoid or the normal chromosome of the bacteria, the such a strain is called as HFR strain. Once the bacteria, those bacteria which have F episome, they form pilus and they transfer this plasmid to the recipient one, which is regarded as female. Once the female receives this plasmid, it also gets converted into male, and the donor while remaining male itself. Then comes the plasma membrane. It is very similar to higher organisms and it follows fluid mosaic model. But certain extra thing which is contained in the plasma membrane of bacteria is coenzyme Q, carotenoids, vitamin K, which play important role in the electron transport chain of bacteria. Because this plasma membrane and its modification, which is called as mesosome, functions as mitochondria of higher organisms. The function of plasma membrane is it is the site of nucleoid attachment. That means that their genetic material remains attached to the plasma membrane, which provides site for its attachment. It doesn't afloat in the cytosol. The plasma membrane is the site of oxidative phosphorylation because it contains the enzymes for electron transport chain. Its enzyme catalyzes production of polymers of cell wall. That means it is important in formation of cell wall. The plasma membrane also acts as osmotic barrier along with the cell wall. The mesosome, which is the modification of plasma membrane, is the infolding of plasma membrane, and it performs the function very similar to the mitochondria of higher organisms. The mesosome, they play important role in replication of DNA, and they also play important role in formation of septum and all the enzymes which conduct the electron transport chain. They remain laid in the mesosomes. Now come to the cell wall. Cell wall is a rigid, dense layer around the plasma membrane, and it protects from the higher osmotic pressure difference created due to the environment in which bacteria are placed. It is made up of peptidoglycan, which is murin, mucopeptide, and glycopeptide, has a polysaccharidic backbone and consists of alternating muramic acid and glucosamine. Precursor of peptidoglycan is diaminopimalic acid. When bacteria is treated with lysozyme or autolysins, they become wall-less, and such wall-less forms of bacteria are called as L forms. But later they regenerate their cell wall and becomes the normal forms. Lysozyme, as you know, is present in our saliva. We have it in our tears and also in sweat. Now the last structure and the outermost structure is capsule, which surrounds the cell wall, and it is made up of polysaccharides. When only one type of sugar is making up the capsule, it is called as homopolysaccharide, whereas when two types of sugars are present, it is such a capsule is called as heteropolysaccharidic capsule. The function of capsule is to protect against the attack of phagocytes, and it also protects the bacteria from rapid loss or gain of water. So dear students, I think this is all in this lecture, and in the next lecture we'll study the remaining part of the structural organization of bacteria, and we'll also take up the difference between gram positive and gram negative bacteria. So thank you very much.